I would like to ask now Peter Mouritzen to unmute his microphone. Ah, he's there. And Peter is representing Samplix. And Samplix is a company which is based in Denmark and introduced in 2012. Very interesting company. They are focusing on the drop instrument and droplet technology. And Peter Mouritzen is the Vice President of Application and Market Development. Peter, you can take it over, please, and share your presentation with us.
Thank you very much.
Okay, that looks good. If you can just enlarge that, then we are set. Perfect.
Like this. Okay, great. Thank you for the kind words and also for inviting us to speak here today. As Udo said, I'll basically stay within the droplet topic, but a slightly different angle. We have a technology at Samplix called Xdrop, which we apply to do targeted enrichment of genomic regions, which are compatible with both long and short read sequencing, and that's what I wanted to talk about today. So in doing that, I would like to just touch upon the principles of the technology as well as the workflow, and then share a few applications with you. So on the right side here, the picture is basically showing the XdropTM technology, the system. With the instrument here in the middle, which is a droplet generator, and then the various reagents that goes along with that instrument. So there are two different cartridges that can make different types of droplets. And then there are various reagents that go into the cartridges to make the droplets, and also that goes within the droplets to carry out various reactions. So Samplix is both a solution provider of this, as well as offering services on this platform. So let me talk a little bit about the principles and also the workflow. So most people are familiar with normal capture technologies like amplicon-based capture and hybridization-based capture, and so in order to do this type of capture, you would typically need to know the region that you want to sequence or capture, followed by sequencing, and then you would simply tile your capture probes or PCR amplicons across that region. So the Xdrop system is different, and it's different because you basically carry out what we call indirect sequence capture, and you will need to design a short 150 base pair amplicon. It doesn't have to be in the middle of the target, but can be some distance away, and hence the name indirect sequence capture. So this 150 base pair amplicon is basically all you need to be able to enrich 100 kb region, and you just need 10 nanograms to start the whole thing. So let me talk a little bit about the workflow. So you will start with high molecular weight DNA, and you will mix that with primers against that short amplicon, which we, by the way, call the detection sequence. So you mix the two, and then you create droplets within this cartridge up here. We generate around 8 to 9 million droplets, and so the concept here is basically to partition the genome to the state, to a condition where you have very few long DNA fragments per droplet, ideally just one. And then you bring the PCR primer set in action to generate the amplicon of the detection sequence. And after you've done that, you will basically know which of these droplets contain long DNA fragments from the region of interest, and those that don't. And at this point, you will simply put the droplets into a regular cell sorter, which you normally use for sorting cells, and that will allow you to get rid of all the negative droplets that contain you could call it off-target DNA, so not related to your region of interest. So you harvest the DNA from these green droplets, and then you create another set of droplets. You partition again the DNA that you harvested from these droplets, and you partition that into a new set of droplets to carry out multiple displacement amplification. And you typically obtain enrichment of around 1,000 times when doing so. And the idea about putting those DNAs into droplets and then doing the amplification within droplets is basically that this is known to be a very unbiased way of amplifying DNA. So it also means that you can amplify and sequence GC-rich regions, repeat regions, and things that are normally considered to be complicated to amplify. So in order to run an experiment like this, you need this detection assay, and we would typically design the detection assay somewhere within the middle of the region of interest, and then we will utilize this to pull out long fragments that all have in common that they contain this detection sequence. And again, we would also include probably an evaluation sequence in the vicinity of that, so allowing us to run a PCR to see how much enrichment did we obtain before we actually move on to doing the sequencing itself. And again, typically we obtain 1,000 times enrichment. If you are working with a unknown region of interest, so basically the orange bit here in the middle is a gap and you don't know what's in there, you would place the detection sequence on the sides of this and then fish out long DNAs that bridge the gap, and then you can sequence this and get to know what is within the gap. So what does that typically look like? So if we have designed a detection assay and pull out DNA and then do here, for example, Oxford nanopore sequencing, you get coverage across a region, typically up to 100 kb, sometimes a bit more and sometimes a bit less. And you will see a pattern where you have the highest coverage in the middle, where you have the detection sequence, and then it kind of levels off towards the ends of these 100 kbs. And then, of course, if you zoom in into just a 10 kb window, you'll see that you have hundreds of times of coverage within that region. So let me talk about a few applications. So I said that you can also enrich regions with a high GC content, and then you can sequence those. And this is an example of this. So here we have looked at the Epstein-Barr virus, which contains a region here marked by the red square along this circular map of the Epstein-Barr genome. And this particular region here is basically a 1.5 kb region which contains 125 base pair repeats, very GC rich, 20 of those altogether, making up these 1.5 kb. And the coverage that you obtain here, so again, we place the detection sequence, as you can see down here, a little bit away from that area because the normal PCR amplicon would probably not run within that area there. But when you do that, you can basically pull out DNA and sequence this. Here we've used PacBio sequencing and been able to obtain, I think it's around 50 times coverage across this region here, which is very GC rich. You can see a GC plot down here, so we are well above 75% and sometimes up to 90 in that region there. So an example of how you can utilize this indirect sequence capture to really also get to regions that are difficult to handle normally. We're also applying the XdropTM technology for validating Crispr editing. And I just wanted to spend a small word about the Crispr editing. So what we typically analyze is unintended on-target editing. So meaning something that happens in the vicinity of where you have brought your Cas9 nuclease into action, creating a double-strand break, because this is where you typically observe indels, and with indels that are most frequently occurring close to this double-strand cut site, and then becoming less and less as you move away at a distance from the cut site. The indels basically occur independently of which CRISPR system you apply, if you deliver by plasmid or as protein. And it also happens independently of the pathway that is working to repair this double-strand break that Cas9 generates. So the way we utilize Xdrop here is that we simply place the detection sequence at a distance away from the double-strand cut site, and kind of avoiding the area where there is an increased risk of seeing indels that might impact this small amplicon by simply removing one of the primer binding sites. And then we'll pull out long fragments again, typically again, and seeing enrichment around 1,000 times. Then having coverage across a 60 to 100 kb region. And then we just simply start looking for loss of heterozygosity, looking upstream and downstream here, and the loss of heterozygosity, of course, being indications of a deletion in that area. So we have applied that to a set of cell lines. It's an isogenic Alzheimer's disease model consisting of 5 cell lines, in which 4 of those have been modified at 2 SNP positions in exon 4. The creation of these cell lines was described in this paper in Stem Cell. It's a very thorough paper in describing both how this was done and also how the validation was performed. And the validation was performed in 6 different kind of independent methods. One of those being creating a short PCR amplicon across these 2 SNPs and then sequencing it by Sanger. Unfortunately, this approach will fail when you have deletions, and that's probably also what had happened here, because we were able, by applying the indirect sequence capture, to show that actually 3 of these 4 edited cell lines actually didn't receive the editing that was intended. And the reason for that was that plasmids appear to have integrated and impacting the generation of the amplicon that would have been Sanger sequenced. So indirect sequence capture is definitely an advantage when you are looking at unintended editing at the on-target site. And if you want to read more about this, there is a bioRxiv paper out by Blundell describing this in more detail. So the last example I wanted to discuss here is how to identify insertions, transgenic insertions, and we've done that in an NCI transgenic mouse cell line. And of course, here we design the detection sequence within the construct itself, and then we are hoping to pull out long DNA fragments that kind of bridge across the breakpoint between the construct insert and then the genome, and then sequence that, of course. And in doing so, we're able to, and then analyzing the reads, we basically first map to the construct, and then we see all those reads that map to the construct, which of those do also map then to the mouse genome. And we did that, and we're able to show that on chromosome 1, there is a set of borders to be found. There's also almost 100 kb deletion, but this is indeed where the insert is located. There are some additional peaks on chromosome 2, but that's part of the construct that was made. It contains PAX8, which is a mouse promoter of PAX8, which is basically located on chromosome 2. There's a small region on chromosome 4 also, which have homology to the flanking arms of the construct. And then part of the construct contains human estrogen receptor. Mouse has an homolog or paralog on chromosome 10, where we also get a signal. And so then in the end, we're able to resolve basically the entire integration of this construct. So it's actually turns out that it's a tandem insertion located on chromosome. So we have more applications. You'll find those on samplix.com. There are some applications explaining about how we can do phasing. Also, there is a bit about from the plant world where it has been applied to close gaps, and also to look at gene clusters and so on. So with that, I would like to summarize. So So the Xdrop system is really about being able to work with very low amounts of DNA. We just need 10 nanograms to do an enrichment. You just need to be able to design a 150 base pair amplicon, and that makes it very flexible. You can also access unknown regions and regions with high TC content, and you will typically capture around 100 kb, but we have seen up to 150 kb. So this is a targeted approach, of course, and which makes it affordable, and it's compatible with both long and short read sequencing. And so that brings me to the very end, and I would just like to acknowledge our collaborators at SciLife Lab and at NCI and Bioneer. So thank you very much.
Okay, Peter, thanks a lot for this talk. I do have a question here because you mentioned in your abstract that it only requires 10 nanogram of DNA. The question is, what are you going to recommend to quality control the DNA? Are there any suggestions from your side? Is that fluorescence identification or some sort of looking more in depth into the DNA?
So we normally just run a tape station or a bioanalyzer chip to see that you have high molecular weight DNA. It's not that we need super high molecular weight DNA. It's just something that is 40 to 50 kb is fine to work with. You can certainly work with something that is higher molecular weight than that. And you can also work with something that is lower, but the region that you will enrich is kind of correlated with the input DNA. So if you have a very short DNA that you input, you'll not be able to fish up long fragments, and that will not give you the breadth of coverage, kind of.
Mm-hmm. Mm-hmm. I mean, you set up a fairly new company, Samplix in Denmark. We know that we do have some issues in getting new biotech companies funded in Germany. How is it in Denmark? Had you had problems, or did you get enough support?
So far this has been fine. I think what you're referring to, I think, is a European issue, which is different from the American situation, as far as I understand. But so far we've been good with private investors.
Okay, good to know, and good luck also for the future.